Dear colleagues, while studying the diabetes mellitus or the anti-diabetic drugs, it is useful to understand the physiology of glucose transport. The objective of this presentation are to update the knowledge about physiology of glucose transport by two different transporter families, to appreciate the role of glucose transporter dysfunction in causing certain disease states, and to understand the mechanism of action of some of the anti-diabetic agents and emerging therapies with glucose transporters, and to explain the rationale of use of salt and glucose in diarrhea. There are two types of the transporters found on the cell membranes in our body. Glucose as such is not able to get into the cell because it's a polar compound. Therefore, it requires help of the transporter. Transporter proteins are of two types: sodium glucose linked symporter or co-transporter that carries glucose along with sodium across the cell membrane, and this is controlled by SLC gene. The other one is facilitative diffusion glucose transporter, we call it as GLUT, and that is controlled by SLC2 gene. If we compare these two, both are transmembrane proteins, and structure of GLUT is 12 membrane spanning helices. Structure of SGLT is 60 to 80 kilodalton, 14 transmembrane helices. There are three classes of GLUTs known today, and there are 6 types of the SGLTs known today. The role of the GLUT transporter is to facilitate diffusion of glucose in and out across the cell membrane, whereas SGLT also transports glucose, but as secondary active transport does not use ATP directly, but relies on the gradient created by sodium potassium ATPase. Cellular location of GLUTs is, or many cells have GLUT proteins on the basolateral and apical membranes, whereas most of the SGLT transporter is located on apical membrane, the cells of the intestine, kidney, and many other organs, including pancreas. Now, as discussed earlier, there are three broad classes of the GLUTs. Class 1 is a group where glycosylation occurs on the loop 1, and the main function is glucose transport. Its subdivisions are GLUT1, GLUT2, GLUT3, GLUT4, and GLUT14. GLUT1 is located on beta cells and liver. GLUT2 is located on beta cells of the pancreas, liver, and kidneys, and particularly located on basolateral membranes of enterocyte, and is responsible for major bulk of the transport of glucose during food intake. GLUT3 is a neuronal one, and that provides the glucose to the neurons for appropriate function. It is also expressed in tumor cells. GLUT4 is insulin sensitive GLUT, and therefore, depending on the under the influence of insulin, it transports glucose. Insulin actually translocates GLUT4 from vesicles to plasma membrane, where it allows glucose to enter. GLUT14 is a type of GLUT3 and is found in testes. Among the class 2 of the GLUTs, the main function is transport of fructose in addition to glucose. GLUT5, GLUT7, GLUT9, GLUT11, and subtypes of GLUT11 A, B, C. GLUT5 is located on the small intestinal cells, particularly apical membrane, testes, and kidney. GLUT7 is also located on colon and prostate. GLUT9 is located on kidney, liver, placenta, and GLUT11 on many cells. The main function is transport of the fructose and also has affinity for glucose. GLUT9 is high capacity uric acid transporters. Class 3 GLUTs are the ones where glycosylation occurs on the loop 9 instead of loop 1 for class 2 and class 1. The subtypes include 6 GLUT 6, GLUT 8, GLUT 10, GLUT 12, GLUT 13. Their location is also characterized brain, spleen, white cells. GLUT6 functions as a glycolytic modulator in inflammatory cells. So it is also involved in lysosomal transporter of glucose during inflammation. GLUT8 is testicular germinal cells and brain, and it transports the glucose into intracellular organelles, that is mitochondrial lysosomes and endoplasmic reticulum. GLUT10 is also located in many cells. It's involved in mitochondrial function and integrity of vascular intima. GLUT12 is located on fat cells, small intestinal cells, muscle cells, placenta. It now transports D-glucose and deoxyglucose. It is also insulin sensitive like GLUT4. GLUT13 is also known as hydrogen-driven myoinositol transporter (HMIT). It's located in fat cells, kidney, brain, particularly hippocampus and hypothalamus, and it transports IP3 and therefore helps in formation of phosphatidyl inositol in brain. Now, the alteration in the blood transporters in drug diseases and by drugs. Some of the examples I am citing here that science is still evolving. GLUT1 is overexpressed in squamous cell carcinoma of lung. Exercise promotes GLUT exocytosis. GLUT2 gene mutation causes severe Fanconi-Bickel syndrome. GLUT4 expression is disrupted in type 2 diabetes. It is said to be downregulated and it contributes to insulin resistance. Metformin ameliorates insulin resistance by inducing GLUT4 translocation, and so does to some extent pioglitazone. Tri- Trilobactam, a natural food additive found from lithocarpus leaves, has been shown to lower glucose through GLUT2 overexpression in diabetic mice. Indinavir, which is a protease inhibitor, inhibits GLUT4. Antipsychotic drug olanzapine inhibits GLUT4 and may result in hyperglycemia and metabolic syndrome. Plant-derived cytochalasin A inhibits GLUT4. Floretin also inhibits GLUT1. The role of GLUTs is being thus evolved and more is being researched to find out its role in causation of dementia, inflammatory bowel disease, and malignancies. A number of inhibitors are also being developed for the treatment of solid tumors and multiple myeloma. KL11743 is one such broad spectrum GLUT1 which is undergoing intense investigation in malignancy. Just like GLUTs, the second group is the sodium glucose co-transporter SGLTs. Now there are six types of the SGLTs and type 1 and type 2. This is SGLT1 and SGLT2 are very important so far as glucose transport is involved in intestine and kidney. Apical membrane small intestine including L cells and K cells which secrete GLP and GIP have SGLT1 receptors also. Now here the main function is that SGLT1 has high affinity but low capacity for glucose transport, and absorption of glucose from intestine and stimulation of GLP-1 secretion are the main functions of SGLT1. In contrast, SGLT2 is found in proximal convoluted tubules of that is segment 1 and segment 2. It has a low affinity but high capacity. Therefore, it is involved in reabsorption of the bulk of glucose from glomerular filtrate. And it is said that 90% is reabsorbed by the SGLT2, and remaining 3 to 10% is absorbed by SGLT1. SGLT3 is found in intestine, testes, uterus, lung, brain, thyroid, and it acts rather as a not as a transporter but sugar sensor. Therefore, it regulates the sugar levels in intestine and brain. SGLT4 is also found in many organs, including uterus and pancreas. It has a low affinity, and absorption of mannose, fructose, and glucose can occur through its activity. GLUT5 is found mainly in renal cortex, is a high affinity transporter for mannose and glucose, and also it transports alpha methyl D glucose. SGLT6 is found in CNS, renal cortex, intestine, and it also has high affinity for myoinositol, and therefore it is involved in maintaining osmotic pressure. Now, in diarrhea, SGLT1 activity is intact in secretory diarrhea, and glucose is absorbed along with two sodium ions of the ORS through activity of SGLT1, and water follows along by osmosis. Therefore, it is estimated that if ORS is not given, fluid replacement will be difficult and several liters of fluid would be required. And therefore, is the role of SGLT1 in treatment of diarrhea with ORS. Now, targeting intestinal SGLT1 and developing its inhibitors. The major glucose absorption of the apical breast border of the intestine is by SGLT1, and glucose from enterocyte transported to the blood from basolateral membrane by facilitative GLUT2 protein. Others are also involved, but it's mainly GLUT2. SGLT1 is also involved in secretion of incretins, that is GIP and GLP1, and other gut hormones related to glucose homeostasis. Even its role as an immunological protective mechanism has also been proposed, and this may be in the treatment in the gastrointestinal infections which are mediated through interleukin 10. Now, in view of these facts, selective SGLT1 inhibitors are being developed. T1095 was found to be highly selective inhibitor, which was a prodrug and was converted into intermediate T1095A, and that also inhibits SGLT2. However, this compound was not developed further because of safety issues. Now, inhibition of SGLT1 provides benefits that includes tissue protection, particularly cardiac protection and renal protection. SGLT1 is overexpressed in ischemia and hypertrophy. SGLT1 might generate reactive oxygen species which promote cardiac dysfunction and remodeling. And because there is a direct reduction in glucose absorption, an indirect increase in the incretins, that is the insulinotropic effect. Now, enhancement of SGLT2 inhibitor action has also been found in the presence of SGLT1 inhibition. And in fact, later on we will learn that dual inhibitors are also being developed. Now, licoflozin is a dual inhibitor, still an investigational drug for the treatment of obesity and polycystic ovarian disease. Sotagliflozin is a dual inhibitor with a high ratio, that is SGLT1 to SGLT2 ratio 1 is to 20, and is under FDA approval process for the treatment of type 1 diabetes mellitus. Mesagliflozin is highly selective SGLT1 inhibitor with higher selectivity for SGLT1 300 is to 1. But this is not absorbed adequately when given orally. Therefore, it has only a local action. So it has been purported to be used in constipation. Now, now we can understand that glucose transporters, glucose homeostasis and diabetes are interrelated things. Glut and SGLT play a role in glucose transporter. Reabsorption of water in salivary ducts depends on SGLT1 activity. An increased SGLT1 expression is found in the salivary ductal cells in diabetes and hypertension. This increased expression reduces salivary flow in diabetes mellitus and also reduces salivary flow in hypertension. SGLT1 activity is increased 3 times in intestinal blood border cells in diabetes, and also type 2 diabetes GLUT5 activity is increased in skeletal muscles and decrease in fat cells. Pioglitazone reverses this trend. Obesity and hypertension increase GLUT5 activity. And the gene for GLUT10 is located on the same chromosome, which is the locus for type 2 diabetes mellitus. Now, these salient facts would stimulate us to find out more about the relationship between SGLT transporters and diabetes. Insulin stimulates LGLT1 activity mediated through protein kinase C activation, which in turn leads to recruitment of SGLT1 to the plasma membrane. This enhances tissue uptake of glucose. Genetic mutation in SGLT1 causes glucose galactose malabsorption. In diabetes, activity of SGLT1 is increased threefold in intestinal cells. And cardiac sarcolimal SGLT1 is increased in type 2 diabetes and decrease in type 1 diabetes mellitus. Increased SGLT1 activity causes cardiac uptake of glucose in diabetes mellitus. SGLT2 gene variation may cause syndrome characterized by tortuosity of arteries, elongated phase, and expression of TGF beta 1. This is called as arterial tortuosity syndrome. Angiotensin 2 is shown to induce SGLT2 production via AT1 receptors. Therefore, there could be an interrelationship between renin-angiotensin excess and SGLT1 or 2 expression. The flavonoid fluorescein, isolated from the bark of apple trees, is a natural inhibitor of both enzymes. Although it is too early to draw conclusions, studies have shown that SGLT2 inhibitors dapagliflozin and empagliflozin reduce insulin requirement in type 1 diabetes mellitus. It is important to realize that dapagliflozin and empagliflozin are well-established drugs for the treatment of majority onset diabetes, that is type 2 diabetes mellitus. Sotagliflozin, which is a dual inhibitor, was found to reduce weight, hyperglycemia, levels of hemoglobin A1C, and insulin requirement in juvenile diabetes, type 1 diabetes. A potential risk is development of ketoacidosis when these agents are used, and more is being understood about use of SGLT inhibitors in diabetes mellitus juvenile type. Now, inhibitors of SGLT2 are canagliflozin, dapagliflozin, altugliflozin, empagliflozin, epagliflozin, lucigliflozin, remogliflozin, tofagliflozin. Now, among these, tofagliflozin is the newest one with high selectivity for SGLT2, whereas canagliflozin has a low activity for SGLT2. Earlier, we studied that sotagliflozin and mesogliflozin are more selective for SGLT1 than SGLT2. In obesity, even without diabetes mellitus, there are studies going on with the role of SGLT2 inhibitors. Particularly, weight reduction is expected when there is glucosuria with SGLT2 inhibitors. And it is estimated that with the use of dapagliflozin type of drugs, there is loss of 60 to 100 grams of glucose per day. Meta-analysis of the studies show that an average weight loss of 1.5 to 2 kilogram will be achieved with the use of any of the SGLT2 inhibitors which we discussed earlier when used as single drug. However, when used with other appetite suppressants or central acting drugs, weight loss would be greater. For example, canagliflozin with phentermine and canagliflozin with other central acting appetite suppressants reduce weight by to the tune of 2.4 kg in 24 weeks to 7.3 kg in 26 weeks. So there is a definitive role of SGLT inhibitors in management of diabetes, although there is a modest reduction in the weight. Now, SGLT2 inhibitors promote lipolysis in rats. It is the mechanism how they reduce weight. They reduce glucose and they increase glucose excretion. And in obese individuals, plasma glucose, insulin, and increased glucagon levels under fasting and postprandial conditions have been found. Now, they reduce portal blood insulin glucagon ratio that promotes lipolysis and provides fatty acid as a fuel for generation of energy. And during this process, they also generate ketone bodies, and this condition resembles metabolic status during fasting. And this is how there is a reduction in the fat in adipose tissue and promotion of lipolysis. And further, SGLT2 inhibitors may reduce adipose tissue inflammation, which is found to occur in obese adipocytes. So the key points of this discussion are: number one, SGLT1 and SGLT2 are the main apical membrane glucose transporters in cells of the gut, kidney, and other organs. GLUTs are the main facilitator glucose transporters, basolateral membranes of the cells. Transporters are critical in maintenance of glucose homeostasis, and this transporter function is disturbed in diseases such as diabetes mellitus, or there may be some gene mutation that may result in rare syndromes. There is a role of transporters in health and disease, and some diseases, particularly diabetes mellitus, may show changes in the GLUT expression. SGLT2 inhibitors have found a firm place in the management of type 2 diabetes and being tried in type 1 diabetes. And development of SGLT1 and dual inhibitors is underway. And next time we may find drugs which inhibit not only SGLT1 but also SGLT2. So there is reduced glucose absorption and increased glucose excretion, and may find a better drug for diabetes mellitus. These are the key references you can access, and thank you very much for the patient hearing.